All right, let's talk about the enzymes, also known as the helper proteins. I don't know why I put helper in quotes because I wasn't quoting anybody, and they actually are helper proteins, so not quite sure where that one came from. Anywho, okay, so life is built on chemical reactions. We would not be here, you would not be listening to the blissful sounds of my Midwestern accent right now if we didn't have chemical reactions. So chemical reactions are essentially the foundation of living, how life is allowed to facilitate, how we're allowed to propagate the species, how we can do all those amazing things that we do. So you're looking at the building of molecules, okay? We know what molecules are. We know what those macromolecules are. We know about carbohydrates, lipids, proteins, nucleic acids. Now, enzymes help facilitate that, right? They're kind of, like I said, the party accelerators. If you're at a party and all the dudes are on one side and the ladies are on another, and because you're too scared to talk to each other, enzyme is going to be that cool person that comes in and kind of gets things going. So they do things by building up molecules and also breaking down molecules. So not only is your enzyme your party accelerator that might introduce you to, your next boyfriend or girlfriend, but they're also the ones that are going to be like, hey, buddy, you can do better. That's what enzymes are, like your best friend that you sometimes need a break from because they're a little bit too intense. But for the most part, they're the ones that are kind of like looking out for you. Okay. So nothing works without enzymes, okay? Every reaction needs an enzyme to work. So you've got your two major types, your synthesis and your digestive. Synthesis, bring them together. Digestive, break them apart. Makes sense. They also speed up reactions, right? They can help to facilitate, but they also can speed up those catalysts. They're the ones that can kind of get things going. So here's your example, right? That kind of like blobola thing right there, that tan blob thing, it kind of combines with two enzymes and the enzyme combined with two molecules links them up. They also, like I said, can break them apart. But the thing I want you to notice is that entire enzyme kind of has to bond with those two molecules so that it's either bringing together or tearing apart. So they're rather large substances. They're also proteins, right? So each enzyme is designed for a specific reaction. They're very specific. Each enzyme needs to be the right shape of the job. Remember, we talked about how proteins, their function depends on their shape. So enzymes, which are proteins, need to have the right shape for the job to do, right? And they're also named for the reaction. You've got sucrase, that breaks down sucrose. Proteases break down proteins. Lipases, you see where I'm going with this, guys? Break down lipids. Polymerase then builds up DNA. So if you're thinking what I'm thinking, you're like, oh, enzymes generally end in ASE. You would be correct. Here's another cool thing about enzymes is that they're not used up. Once they're not like a mosquito that stings you and dies, okay? They can be reused for the same reaction with other molecules. And a lot of times you need a very little amount of enzymes to help in many reactions. When I had you guys spit in the test tubes, many of you spit a fair amount, which was good because we wanted to make sure we had ample saliva to conduct the experiment, but you really don't need that much. Enzymes, you don't really need all that much to get that party started. Again, it's the shape that matters. So a couple things I want to some terms. I don't know, you guys love vocabulary, so this slide's dedicated to you. You have your substrate, which is the substance that's broken down. So in this case, in this example, it's the sucrose. In our lab, it was the starch. The substrate is a substance that's being broken down, okay? The enzyme, or building up, depending on. The enzyme is, so for example, this is a digestive. This is an example of a digestive enzyme. So it is being broken down. So your substrate, your sucrose, is being broken down. Your enzyme does it. Your enzyme actually physically breaks it apart or sticks them together. And if you look, that active site, another vocab term, is very specific for that substrate, for that sucrose. So this sucrase is built for the sucrose. So I'm just letting you know, don't bring your sucrase to a protease party, because everyone's going to make fun of you, okay? Write that down. So again, what happens is that it builds, it breaks it apart, and then you have down there, you've got your glucose and your fructose. The products are released. You also have some water that you have to add, and also these take energy. These reactions take energy. There's a bigger version of this, kind of the same thing. It's amazing, wonderment of life. Again, you guys love vocab, so this is for you. It's your helper protein. And then your substrate's the molecule that works on the protein. The products that is produced, either you're going to have 2 at the end if it's a digestive, or you're going to have 1 at the end if it's a synthesis reaction. And then your active site, that's where the part of the protein bonds with the substrate. So a couple things. What are some things that can affect enzymes' ability to do their job? The correct protein structure, right? Or in this case, to go a little further, it's the correct order of amino acids. Remember, shape dictates function in proteins. Temperature, right? Why does it say, so letter made a boo-boo. It says why enzyme has to be the right shape. Under temperature, it did say why, because if you want, for many cases, if you want to increase enzyme function, you can increase the temperature. But if you go too hot, you could also denature it and kill it all off. So ignore what I have there. Again, pH has to be right too. And we saw that with your lab. HCl, which is a stomach, very similar to your stomach proteins, or your stomach acids. We saw that if you put it in a low pH, so an area of high acidity, there you go, it's not going to work. So enzymes are they're great party accelerators. They're there for you, but they're kind of finicky. So you have to make sure you've got the right snacks, you've got the right DJ, and you have the right temperature, because that enzyme is not walking in to a basement party that is way too hot. Nuh-uh. Or too cold. Figure it out, right? So there's another part of this that we can talk about in class, which is the exergonic and the endergonic. So that's kind of the reactions that take energy and the reactions that release energy. And I'm sure you can imagine the synthesis is going to take energy to put those molecules together, and the digestive is going to is going to release energy as it breaks apart. So that's all I got. Just giving you some information about enzymes and life, because that's what I do, because I'm a wofo. See you guys tomorrow.